Hi everyone. In this video I will talk about Krebs cycle. So as we know, the end product of glycolysis is pyruvate, right? So when pyruvate is reached, there is a reaction which uses pyruvate dehydrogenase complex. So this is the enzyme, pyruvate dehydrogenase complex. And this enzyme would actually convert the pyruvate into acetyl-CoA. And then acetyl-CoA could actually enter the Krebs cycle. This is a 2-carbon molecule. So it would actually enter the Krebs, the inner membrane of the to go into the matrix. And this whole process is happening on the mitochondria. So this is our outer membrane of the mitochondria, and this is the inner membrane. This is the interspace of mitochondria, and this is the matrix, okay, where Krebs cycle take place. So this is the part that I'm zooming in is actually the matrix. So this is where we're going to talk about. So acetyl-CoA enters, and as we know, acetyl-CoA is a 2-carbon molecule. Okay, it's a 2-carbon molecule, and there will be an existing intermediate which is called oxaloacetate. So this intermediate will be already there. It's a 4-carbon molecule, and both of these two would actually combine with the aid of citrate synthase. This is the enzyme responsible for citrate product to produce citrate, right? And as we mentioned, this is a 2-carbon molecule and this is a 4-carbon molecule, so they are actually going to give us a 6-carbon molecule. So citrate is a 6-carbon molecule, and basically this is our first regulatory step here. So it's only going into one direction. So whenever you're having a carbon number change, so we're changing the number of carbons, then this is a regulatory step, okay? And you're gonna see later here in the rest of the steps. So from citrate, isocitrate will be formed with the aid of aconitase. This is the enzyme responsible for this, and it's basically there is water going in and water going out. It's basically the same exact thing, so there is no change in the number of carbons. And then after that, the isocitrate will be converted into alpha ketoglutarate. So in this reaction, alpha ketoglutarate is actually a 4-carbon molecule. And remember, as I said, whenever you're seeing a carbon number change, so the number of the carbon is actually changing, then this is a regulatory step. So the enzyme responsible for this reaction is a regulatory enzyme. So it's only in one direction, okay? So in this reaction, because we lost a carbon here, then there will be a carbon dioxide secreted, and the enzyme responsible for this is isocitrate dehydrogenase. Okay, this is dehydrogenase. Okay, and as I mentioned before, so whenever you're seeing dehydrogenase, then there is a usage of NAD or FAD. So here it will be NAD converted into NADH. This is our first energy molecule in the Krebs cycle here. And so we're done with this. There is a carbon dioxide secreted, there is NADH, and it's a 4-carbon molecule. Right now, we're gonna convert that alpha ketoglutarate into succinyl CoA. CoA. So in this reaction, we're gonna need CoSH, CoASH, and in this reaction, the enzyme responsible for this, and also, as I said, so, I mean, no, wait a second, man. This is a 5-carbon, I'm sorry. This is a 5-carbon molecule. So, from a 6-carbon to a 5-carbon molecule. Okay? And succinyl CoA is actually a 4-carbon molecule. So, as I said, we lost a carbon, then there is a release of carbon dioxide. And the enzyme responsible for this is alpha ketoglutarate dehydrogenase. Okay? So, and this is our third regulatory enzyme. Isocitrate is an isocitrate dehydrogenase is also one, and citrate synthase. So 1, 2, 3. These are regulatory enzymes, and it's only going into one direction. Only going in one direction. And as I said, whenever you see the change in the carbon number, then this step will be regulatory. And also dehydrogenase means NAD to NADH. This is the second NADH here. So right now, we're gonna convert succinyl CoA into succinate. Okay. The enzyme responsible for this will be thiokinase, succinyl CoA thiokinase. Okay, and there will be a release of CoASH, and in this process, it's a unique process because it uses GDP. So it uses GDP and turns it into GTP. This is an energy molecule also. So right now we have succinate. We're gonna convert that into fumarate using the enzyme succinate dehydrogenase. Succinate dehydrogenase. So, but this time, this step is unique because it uses FAD. This is the only FAD to FADH in the whole Krebs cycle. And then from fumarate to L-malate, we're going to use fumarate mutase. And in this process, it only puts in water, H2O. And as I said, 4 carbon here, 4 carbon here, 4 carbon here. So all 4 carbon until we reach to oxaloacetate again. And there is no regulatory steps because it's not changing the carbon number. Okay, so we reach it to L-malate. Right now, from L-malate, this is the last intermediate actually. The L-malate is the last intermediate in the Krebs cycle. Okay, from the L-malate to oxaloacetate, we're using malate dehydrogenase, and this is our last NADH. So NAD to NADH. So right now, how many NADH we're having? We're having this is 1 here, 1 is here, 1 here, and where's the other one? This is 2. Okay. This is 3. The last one. Okay. And how many GTP? There is only one GTP. How many FDH? There is only one FDH. So the net we're having here is 1 FADH, 1 GTP, and we have 3 NADH. Okay? If we're gonna do the math, as we mentioned before, 1 FADH is 2 ATP, yields for 2 ATP. GTP is basically ATP, 1 ATP. And NADH yields for 3, so we're having 3 here. 3 times 3 is 9, so 9 ATP. 9 ATP plus 2 is 11, plus 1 is 12. So we're having 12 ATP yield from just one molecule of acetyl-CoA getting into the Krebs cycle. Okay? And that's it for the Krebs cycle.